Here we're looking at a cross-section of one of these little glomerular loops inside the entire glomerulus. So if we cut through one of the little loops, this would represent the lumen of this capillary where the blood is. So blood is in here. Red blood cells, white blood cells, platelets, and blood plasma. What this is showing is the actual filter, the filtering mechanism. So what does this solute have to travel through to get into Bowman's capsule? All right, and what is going to get through and what is not? So what's going to stay in the bloodstream, what gets filtered? Well, the first layer is the actual endothelium. So this layer here with all the little holes represents the actual endothelium of the capillary. So those are cells with little holes or pores. So that's the fenestrated capillary. What that layer does, it prevents big things. All right, so it's going to prevent red blood cells, white blood cells, and platelets from passing through and getting filtered. The next layer, which is called the basal lamina, prevents big proteins from being filtered. So large proteins do not get filtered from the blood. An example of a large protein, a very important one, is albumin. If you recall, albumin is the #1 plasma protein in our blood. Well, that plasma protein should never leave blood. It should not get filtered. It should not be in a tubule. It should not be in urine. And that's an important clinical idea to remember because we actually test for albumin in urine. And if we find it, that typically indicates there's a problem. So there could be inflammation, there could be trauma, there could be problems at the kidney if we find albumin or other protein in urine. Protein should be very, very low in urine. Shouldn't have any protein. Should definitely not have bigger proteins in urine. Then there's a layer called the slit membrane, which is in between these little pedicels of the podocyte. So here's the podocyte cell, and its little cellular extensions help form another layer. This is like a triple layer filter. And those little pedicels and slit membrane prevents medium-sized proteins from being filtered. So all this stuff, the cells, the large proteins, and the medium proteins stay in the blood. They do not get filtered. Everything else can be filtered. So what would be that everything else? It would be water. It would be ions. All the different ions are really small. They get filtered. Amino acids get filtered because they're small. Glucose gets filtered because it's relatively small. And other small molecules like waste and other things. So those things are important, right? Sodium's important, calcium's important, glucose is important. So we have to have that next step, reabsorption, to start to reclaim all the important things, leave the waste so that can be eliminated. This picture we've seen in the past. We just didn't add all these pressures to it. But these pressures are very similar to what we did back when we learned about capillary exchange. The difference is there's no exchange occurring here. Only filtration occurs at a glomerulus. Because if we add up all the pressures at work, you're always going to have a net filtration pressure that's positive. It's usually around 10. So remember what we did during capillary exchange. We just added up the pressures that are pushing out, like blood hydrostatic pressure. Subtract all the pressures pushing in, like blood colloid osmotic pressure, and you ultimately get net. We're going to keep it fairly simple and make sure we know that it's always a net positive. There's always more pressure pushing out than there is pulling back in. Now, will that 10 go up and down? A little bit. If blood pressure goes up, that's going to go up. But if net filtration pressure goes too high, that's a problem because we're going to filter too much. If it gets too low, we're not going to filter enough. So the idea of this slide is to just know some of the ways we can regulate filtration. One way is using the nervous system. And the nervous system regulation revolves around this guy right here, this arteriole. Notice it shows, this is nerve innervation. The idea here is if we need to decrease filtration, we vasoconstrict the afferent arteriole. Because if less blood enters the glomerulus, less filtrate gets produced. That's a good rule of kidney function. Less blood going into a kidney, less filtration, less urine production. So sympathetic stimulation constricts the afferent arteriole to decrease glomerular filtration rate, or GFR. If sympathetic stimulation decreases, for example during rest, then this afferent arteriole dilates, more blood enters, more filtrate gets produced, more urine gets produced. So this is one of the reasons why the kidneys get more blood at rest and less blood when you're more active. If you think about when you're really active, like exercising, you have a lot of sympathetic activity when you're exercising. So all these little blood vessels going to all these little nephrons are fairly constricted. That's why you typically don't make a lot of urine when you're exercising. The next two things that regulate filtration are hormones. Angiotensin II acts kind of like sympathetic stimulation. It constricts. Angiotensin II is a vasoconstrictor. We learned about it back in blood vessels. It vasoconstricts a lot. It'll vasoconstrict all kinds of stuff. But here we're talking about the afferent arteriole. It also constricts the efferent, but same idea. You want to think of angiotensin II as very similar to sympathetic stimulation. It helps to decrease GFR. The next one is opposite these other two. Atrial natriuretic peptide increases GFR. So if you recall, this hormone is produced by your heart, your atrium. And what stimulates this production is high volume of blood. So if you think about it, what could be a way to decrease a high volume? Well, you filter fluid out. So you can increase filtration, and that will help you get rid of a high volume. This hormone kind of acts as a diuretic because it can get fluid out of the body. It also affects sodium, so it also puts sodium into urine. If you remember, sodium is Na, right? Na plus. There's Na, natriuretic. It puts sodium into urine, and that helps draw fluid out of the body. So this guy is extremely important to help us eliminate excess fluid from our bloodstream. So it's kind of our natural diuretic. Sometimes it's not enough, so we have to give people a diuretic. And we'll talk more about how those work in later slides. So this is neural regulation and hormonal regulation of filtration. Next thing we're going to do is talk about reabsorption. So remember what reabsorption means. We're taking things from the tubule that might eventually be eliminated, bringing them back into the bloodstream. So it's moving things from the fluid in the lumen into the peritubular capillary. So it's the opposite direction of filtration and secretion. It's done all using transporters. And we're not going to learn all these different transporters. But you'll notice on this drawing, there's one right here called a sodium glucose transporter. Its job is to take a couple sodium ions and a glucose molecule, move them out of the tubule into the cells of the tubule, and then back out of those cells and into the bloodstream. The idea here is you take these guys from here and put them into here. So this is reabsorption of sodium and glucose at the proximal convoluted tubule. But there's all kinds of other transporters. And in fact, most of the reabsorption that occurs happens right away at the proximal convoluted tubule. So for each part of the nephron, I want you to think, what is it known for? What's the PCT known for? What is the proximal convoluted tubule known for? What is the descending limb of the loop known for? What is the ascending limb known for? What is the distal convoluted tubule known for? What is the collecting duct known for? So that's basically the next few slides we're going to do. So what is the PCT known for? It's known for most of the reabsorption. That includes 65% of the water that gets filtered gets reabsorbed right away at the PCT. Glucose and amino acids, they get completely reabsorbed at the PCT. So think about glucose and amino acids. Your body is made to not get rid of them. You could always use them for something. You could always store glucose and amino acids as fat. So the body is built to conserve them, to keep them in. Now, diabetics might have excessive amounts of glucose, and the proximal convoluted tubule just doesn't have enough transporters to reabsorb it all. And what happens is some of that glucose will then spill out into urine. But in normal situations, glucose should not be found in urine because it should have been completely reabsorbed at the PCT. It also reabsorbs other important ions. We saw the sodium over here getting reabsorbed, but chloride will then follow, right? Chloride likes to follow sodium. So if you're moving sodium, that forms what's called an electrical gradient, more positive here. So all you got to do is open up little channels, and chloride's going to follow that sodium. Potassium gets reabsorbed, and some other important ions. There is a little bit of secretion that occurs. So now if you think of over here, you might have some hydrogen ions. You might have some ammonium ions, which are waste. You might have urea, which is waste. We've got transporters that move those guys this way and put it in the lumen. So there's reabsorption and secretion that occurs at the PCT. What happens at the loop of Henle? So now we're looking at this loop. Now we have to think of the loop as two different limbs. You've got the descending limb and the ascending limb. This picture is focusing more on this ascending limb here. There's a part of it called the thick ascending limb, but this is the ascending limb here. What is the loop of Henle known for? Well, the descending limb over here on the picture reabsorbs mostly just water. About 15% more gets reabsorbed at the descending limb. Not very many solutes get reabsorbed at the descending limb. At the ascending limb, it's known more for its solute reabsorption. So all kinds of ions get reabsorbed at the ascending limb. Notice here on this drawing, here is a transporter that moves sodium, chloride, and potassium all at once from the lumen to eventually move them back into the bloodstream. A lot of other ions move as well. So a lot of ions get reabsorbed at the ascending loop of Henle. If you've ever heard of a diuretic known as Lasix or furosemide, that drug blocks reabsorption of sodium, chloride, and potassium at the ascending loop of Henle. It's often referred to as a loop diuretic because it blocks transporters at the loop of Henle. So if you think about it, if you block the reabsorption of sodium chloride and potassium stay in the lumen. What that does is helps keep water in the lumen. And the whole point, the whole purpose of a diuretic is to keep more water in the loop. Therefore, more water goes out the nephron. So if you keep these solutes in here, you'll keep water in here. And more water will stay in this loop. More water will be collected into the collecting duct. More water will be found in urine, and that helps remove fluid. So diuretics often block the removal of solutes and therefore block the retention of water. So we'll talk more about that probably in class when we do this drawing. There's a little bit of secretion that occurs. It secretes some urea. So again, urea is a metabolic waste. If it's in here, we can secrete it into the lumen so we can get rid of it. Remember, anything left in the lumen is going to be going out in urine. What is the distal convoluted tubule known for, and what is the collecting duct known for? We're going to do these two portions of the nephron in the next video.